Hi everyone, I'm Sharzad. I'm a PhD student in Civil and Environmental Engineering Department at University of Maryland. And today I'm going to show you guys how I use PCR, polymerase chain reactions, together with gel electrophoresis to characterize the microbial community in my soil samples. So the first thing is that you have to make sure everything is very clean. So your workspace should be ethanol and everything else that you work with. So we use 70% ethanol to clean the whole workspace, and also like the pipettes and my gloves and the rack and everything that I'm working with. So for PCR, you're gonna need PCR tubes, DNase, RNase free. You're gonna need pipette tips in different sizes, and you're gonna need pipettes, different volumes. You're also gonna need for your reactions, you're gonna need DNA-free water and enzymes and nucleotides. So PCR is basically some kind of DNA synthesis process, right? So you're gonna need enzymes and nucleotides to synthesize DNA and amplify the target DNA. So here I'm using DreamTech, which is a master mix that contains these enzymes and nucleotides and also buffers. I also need primers, reverse and forward primers. These primers are necessary in PCR because they help you, they specify which part of the DNA you want to amplify. So here I have primers that will together target a piece of 16S or RNA gene in the helicocoides. And you also need negative controls. So negative controls are samples that you are 100% sure do not have the piece of gene you're trying to amplify with the primers. For that, I'm going to use DNA-free water, and I'm also going to use E. coli, extracted DNA from E. coli, and I'm sure this doesn't have the 16S from the helicocoides. For a positive control, you're also gonna need a positive control. For positive controls, you're gonna need a sample that you're 100% sure that has that piece of gene you're trying to amplify. So for that, today I'm using extracted DNA from WBC2, which is a mixed culture that contains the helicocoides and some other dechlorinators that we have in this lab. So I could just extract the DNA from that, and I'm pretty sure that the gene I'm looking for is going to be there. I also need like my template DNA. So I have soil samples, extracted DNA from soil samples here. I'm also using these frozen racks to keep everything cool, to make sure to prevent enzyme degradation. So to prevent errors from pipetting, we are gonna combine all the reagents in a 1.5 milliliter DNase RNase free centrifuge tube first, and then from here we're gonna dispense them into these smaller PCR tube. So I have calculated the amount that I need from each of these reagents, like the water, the DreamTech, the primers, that would go into each tube, and the total volume for each PCR tube would be 25 microliter. And I have calculated how much the total amount for all the PCR tubes would be, all the 12 PCR tubes that I'm going to use today. And I'm going to add the total volume of water, DreamTech, and primers into this cocktail here. And then we're going to dispense them. So now I'm going to add water to prepare my cocktail. I have the right volume, the right pipette tip. Then I'm going to add the amount needed for DreamTech. before using the dream tag, we have to first vortex it for a few seconds to make sure that everything is homogenized, but very gently. Then I'm gonna take that. After adding the dream tag, we're gonna continue on ice because the enzymes can degrade pretty fast. So now I'm gonna add the forward and reverse primers to the cocktail. To take the primers, I usually pipe it in and out several times to make sure that everything is homogenized when I'm taking the amount that I need. Then I add it to the cocktail. Again, I pipe it in and out several times to make sure that everything is inside the cocktail, because we're working with very tiny volumes. Do the same for the reverse primer. And now the master mix, or the cocktail, is actually ready for the next step. And now to make sure that everything is completely mixed, I'm gonna vortex for just a few seconds to make sure everything is mixed. And then we're gonna centrifuge for a few seconds to make sure that nothing is on the edges and everything is completely ready. So for balancing, you have to use a counterbalance. So now that the cocktail is ready, I'm gonna transfer 23 microliter of the cocktail into each of these PCR tubes. And then to each of them, I'm gonna add 2 microliter of the template DNA that we have prepared. So now I'm gonna add the 2 microliter of the template DNA to each of the centrifuge tubes. You should make sure that all your microcentrifuge tubes, the PCR tubes, are labeled at this point. So I'm gonna pipe it in and out from the extracted DNA again to make sure that everything is mixed. And then take out the 2 microliter that I need. You should put the pipette tip inside the liquid and then pipe it in and out for a few times again to make sure that everything is out. And we continue for all the samples. You might see some bubbles in your PCR tubes, so you just have to go ahead and hit them gently and try to get rid of these bubbles. And then we're gonna centrifuge them to make sure that all the material is down at the bottom. So this is our PCR thermal cycler. From here, I have to choose the method that I want to run the samples with. So I choose the electrophoresis and the primer that I already have, and here is the method. for that run. Now I'm gonna put in our samples here. This. And now it is ready to be run. And it will ask you if the volume, the total volume is 25 microliter, because this method is for 25 microliter. And you hit OK. And now it starts. It also shows you how the cycles work here. So we just have to wait for 47 minutes, and then it's ready. So for running the gel, we first need gel that is made of agarose, TAE buffer, and ethidium bromide. Ethidium bromide is a dye that can bind to DNA, and it fluoresces under UV light. So we add our PCR products to these wells. I'm gonna take this comb off, and this comb has produced these wells. So in each of these wells, I'm gonna load my PCR products, and they're gonna travel through the gel. So the gel acts as a matrix and a sieve for the DNA to travel through. The shorter the DNA, it will travel faster, and the bigger, longer DNA pieces are gonna travel slower. So I place this gel here and add TAE buffer to the field, up to the field line. So now I want to take off this comb from the gel. And now we can start loading the gel. Now I'm gonna load the ladder. I'm using a 100 base pair ladder. So a ladder acts as a measuring tape or a ruler, and it shows the different sizes of DNA. So this 100 base pair ladder marks every 100 base pair. So it starts from 100, 200, 300, up to 1000 base pair. So when we run the gel, we are able to compare our DNA size with the ladder. I start by loading the ladder to both ends. You have to go gently inside the well and release the liquid from the pipette. I'm loading the first sample. Again, I slowly go into find the well, go a little bit inside, and release the sample. So now I have turned on the power supply that is connected to the cap, and it goes on top of this. And the negatively charged DNA actually moves from the negative side of the gel to the positive. And in the meanwhile, the DNA gets separated, and we can see it under the UV light. So hopefully you can now see two colors separating from each other. On the bottom we have the yellow, and on the top we have a faint blue. And I'm running the gel with the voltage of 100 volt. And depending on your voltage and the gel concentration, the time that you need for the DNA, different DNA sizes to fully separate is different. But with this voltage and this concentration that I'm using, I usually run the gel for 30 to 40 minutes. this is the imaging system that we use. It uses ultraviolet light to capture a picture of our gel. You can open it and put your gel here. I have already done that. And then in this application, in the software Alpha View, if you hit acquire, you'd be able to see the final image of your gel. You're also able to annotate your gel, so make all these labels, which I already have. Now let's take a closer look at this gel. On these both ends, you can see the ladders. They mark 100 base pair, 200 base pair, 300, 400, and up to 1000. This is our first soil sample, the second soil sample, the positive control, the water, and E. coli. You can see there are two bands, one in our first soil sample and the second one in the positive control, right between 200 and 300 base pair. So this band is the right size. Our expected amplicon size was around 240, and these two are almost in the are exactly in the right size. And we were sure that the positive control contains Dehylococoides. So this band shows Dehylococoides. And this also indicates that our first soil sample contained Dehylococoides as well. Our second soil sample, you can see there's no band. So TW4 did not have the Dehylococoides. Dehylococoides was not present here. There's no band in water and also not in the E. coli, which is good, showing that there was no contamination and also no non-specific binding to another bacterial gene. So this is basically one of the ways that we have to find out if a specific piece of gene is present in our samples or not. It can be a soil sample, groundwater, sediment, any kind of DNA that you have. And if this piece of gene is present, maybe we can later on do a qPCR and quantify that.